Hi, everyone. I want to welcome you to our first clinical trial toolkit series sessions for the spring 2024. Today's session is titled Quality versus Quantity, Understanding Experimental Design Principle, presented by Dr. Tyler McGahee. For the audience on Zoom, please write your name and your email address in the chat for attendance purposes. Please indicate CE if you would like to receive continuing education credit. I will follow up via email with a continuing education form that needs to be completed. If you have any question, please feel free to unmute yourself and ask, or you can write it in the chat, and I'll be monitoring the chat throughout the presentation. Dr. Tyler McGahee comes to us from the WVCTSI Biostatistics, Epidemiology, and Research Design Core, the BIRD Core. So his expertise will help us learn how to develop more robust studies for the benefit of patients. He is the research imaging specialist with a particular focus on human neuroimaging. He received his PhD from WVU in neuroscience in the lab of Dr. Victor Fenmore at the Rockefeller Neuroscience Institute. He's also from Virginia and received his bachelor's degree from Hampden-Sydney College. In his free time, when he isn't hanging out with his wife and 2 cats, he is a C3 paramedic. Wow. Welcome, Dr. McGahee, and thank you for presenting today. Without further ado, please welcome Dr. McGahee.
Thank you, Maria. I also want to give a special shout out to both Maria and Debbie Lee, who have helped keep me on track and will help me with the technology and chat as we go. So like Maria said, just some general expectations, Zoom courtesy. Please keep yourself muted unless you actually want to ask a question, but don't be afraid to ask a question. You can either drop it in chat. If you don't feel comfortable sharing with the group, you can message me directly. I think Maria logged me in, so just go ahead and message Maria if you have any questions. With that, we'll get started. So here's a brief overview of what we're going to do today. We'll play a quick icebreaker. We'll go over some of the basic study design principles. We'll do a review game, so you will actually need your cell phone, and you don't hear that much in Zoom meetings. We'll go through some case studies, and there'll be some open discussion for good practices and any questions you might have. So, here are our objectives. Today's kind of an open discussion, hopefully with a lot of discussion on the basic principles of clinical design, especially in terms of clinical trials. I want to work with you all to kind of understand some of the basic terms, kind of get into the nitty-gritty, especially as you all are integral parts of the clinical trials process. So here's our icebreaker, and so you'll have to drop in the chat. On the next sentence, there's going to be on the next slide, there's going to be a sentence. I want you to count the letter F in that sentence and drop your answer in the chat as quickly as you can. Okay, here we go. 3, 2, 1, and go. Again, count as many F's as you can find and drop them in the chat. Okay, a couple more seconds. Okay, we will jump to the next one. So let's figure out what the answer is. It's 6. So kind of the point of this one is it's completely essential for us to pay very close attention to the fine details, especially as we're trying to flesh out our clinical trials. We owe it to our participants to make the most out of their data and limit the burden from having to resample or enroll extra patients. So that was just a quick emphasis there on making sure we pay close attention to the principles of clinical design. So, does anybody want to give me a good definition of experimental design? I'm say feel free to unmute or just drop it in chat. Yeah, so Mary, that's a good point. I did rush you, but it is also it is important to take time to make your decisions. So experimental design is important for a number of different reasons. It's really sitting down and thinking about all of the different things that go into an experiment design before we sit down to write our IRB and things like that. It's very important because it helps us ensure validity of our experiment, that it's easy to repeat and verify the results. Not only a well thought out and well planned experiment is great for collecting data and having good and robust experiments, it also facilitates the regulatory process, getting your IRB and data protection, things like that. And it helps us translate those findings to other studies, which we'll talk about here shortly. And it's good science drives scientific progress. So now we're going to go over some of the basic principles of experimental design. The next couple of slides are going to be a little definitional, definition-esque in quality, but it's going to help us understand some later case studies. So the big first divide we see in our experiments are the difference between observational and experimental studies. So with our observational studies, there's no really direct intervention by the researchers. So this can be primary data collection, cohorts, case studies. So like a great example of this is if the ED gets a great patient that's a great exemplar of a particular injury, they might write up a case study on that patient or a couple patients, but they're not directly manipulating any variables per se with that patient. The other kind of big branch of experiments are the ones that we call experimental or true experiments. These are those traditional experiments that we think of with active changes or manipulations by the investigator. So this might be a new study drug, it might be a new therapy, things like that. These are more resource intensive, but they provide us a little bit more power too, because we're able to directly specify what we're looking for and kind of control things better. The next one, as the title says, that we can break down our experiments by is qualitative versus quantitative. So we see qualitative studies a lot with kind of general population-based studies and a lot out of these kind of social sciences. So these are mostly based around surveys and questions and interviews where you sit down and interact with your participant or your patient and discuss things like, how do you feel after X, Y, and Z? Or how confident do you feel? How stressed do you feel? Things like that. The problem here is they're a little more subjective, but they don't require as many resources. And then the other kind of goes with those traditional experiments that we normally think about. These are things that we have more objective measures. So what is the patient's weight? What is this lab value? What are the different metrics associated with that intervention? So the last kind of two definitions we're going to look at to break apart the different types of experiments are prospective versus retrospective. So we'll start with retrospective because these are easy to do and they're quick, but it's off pre-collected data. So the most common one we see here is going to be chart reviews. So we're looking back in the chart to see what medications most common in patients with hypertension, and with those taking a beta blocker versus a ACE inhibitor, does that make a difference? And then with the prospective, again, this goes back to what kind of pops into your head when you think about an experiment. So we're looking at doing an active intervention or manipulation. These do require more resources because you're actually changing things about maybe the standard of care or your intervention, but they tend to be more precise and you do tend to have a better return on investment because you can kind of narrow down your investigative question. So what are some key concepts that we need to keep in mind when we're going through and designing our clinical trial? Feel free to drop any of these that you can think of really quick in the chat. Okay, we got some good answers there. So here, or I think these are my biggest key experimental design concepts that we're going to go over in detail after this. So talking about randomization of our participants, how to understand and quantify bias, how to develop appropriate controls, doing statistics to figure out exactly how many participants we need in our investigation. Of course, we have to consider the ethics when we're talking about clinical trials, especially with humans and vertebrates. And the last is going to be validity and reproducibility, because what good is our science if it can't be checked and verified? So here's another little interactive bit. This is kind of an interesting thing because we all come from different backgrounds and have different roles. If you want to, go ahead and drop your answers in the chat here to the following sentence. All children are capable of blank, but we cannot expect all students to blank if we don't blank. Again, if you don't want to send that to everybody, you can send that to me personally. But remember, I'm not Tyler right now. I'm actually Malia. Okay. Let's see if we can get another good one. Okay. So I'm going to pick on Nina real quick. So all children are capable of learning, but we cannot expect all students to learn if we don't teach them. Another good example that came in was all children are capable of flourishing, but we cannot expect all students to learn if we don't support them. And so see how we can have a bunch of different responses to the same kind of question. And it's all based on who we are as a person and kind of how we've come up in the world. But the great thing about science is we're able to eliminate a lot of this bias because it kind of doesn't matter who you are or where you are in terms of science, as long as you do good reproducible science. The slides are frozen. Okay. So we'll go over some methods to reduce bias. So we obviously don't want to alter or bias our sciences based on who we are, where we come from. So here's some of the methods that we'll go over to kind of help reduce bias. The big one that's really helpful is randomization, but we'll also talk about appropriate controls, some other techniques like blinding. And this is one of my favorite quotes: proper planning prevents poor performance. So that's why it's very important before we just start running out and collecting data to sit down and really think about what we're going to do. So really quickly, just a review of variables. The typical variable we think of is the independent variable, which is the variable that we purposely manipulate. And our subsequent measures of the changes to the independent variable are our dependent variables. The way I keep these straight is if you're sick and you need an IV, you depend on the IV. So the dependent outcomes are based on changes to the independent variable. And there are some other variables that we need to be aware of that we don't necessarily have control over. So a good one that isn't addressed as much, especially out in the open scientific literature, are confounding variables. A lot of times we report it, but we don't do a very good job of controlling for it. So potential confounding variables may be other comorbidities that your patient population has, or things like age. So obviously, if we're looking at a 25-year-old with hypertension versus a 65-year-old with hypertension, they're obviously going to respond different to different therapeutics. Another thing that necessarily we do a good job of tracking, but not necessarily a good job of thinking about and correcting for in our analyses, are categorical variables. So these are things that we obviously can't change, but they might affect our outcome analyses. So things like a blood type or a disease stage, or even region of the country that your patient's from. So there's just things to keep in mind when we're working. So like I said, we'd go over some of the types of controls that you can use to help reduce bias. The first one is probably the most common. So this is going to be our placebo control. This is where a participant receives a placebo instead of the active treatment. So a lot of times this is an investigational product that we're giving the patient, and they'll send a placebo version, which is almost identical, and it's even difficult for us to tell sometimes without them being labeled. The next one is what we call an active control. So this is also pretty common in our clinical trials group. So a lot of times we'll have a new investigational product and we'll compare it against the current standard of care and see if there's a difference. And the last one that's not used quite as often is a historical control. So this is similar to the active control in that we compare against the standard of care, but it can be based on data previously from like the electronic health records. So for instance, if orthopedic surgery wants to try out a new suturing method to look at infection rates, they can try out the new the new suture, see if it works now and compare it to their previous infection rates over the last year or so. Some other things to consider when we're looking at our controls. When we're giving a placebo, it should be as close to the investigational product or the active product as possible. This helps almost with single blinding. So the participant has a hard time determining what is the investigational product versus the placebo. The other thing that we're pretty good about, but we also need to pay attention to, is matching controls. So we can improve our validity and power in our experiments by having appropriate matching controls. So things like biological sex and age between your experimental and control groups. And the last one is the use of blinding. And so I know there's a couple clinical trials going on right now at the university that are double blind. And so this is where both the participants and the either interventional team or the investigation team don't know which patients receive which product, whether it's the investigational or the placebo. And then that goes back to the sponsor or the principal investigator to look at the data. All of these will help you add power to your experiment. The other big thing that I know we cover in things like CITI, but we don't think about as much as we should when we're designing our experiments, are the ethical principles. So things like informed consent, respect for our participants and patients, benefits, justice, and non-malfeasance. So I know we all talk about informed consent, and we think about the informed consent form. That's this 20-page document that I have to submit to the IRB, but it's kind of nice sometimes, especially when you're trying to protect your patient or participant, to take a step back and think about what are the principles of informed consent. It's kind of an ongoing process throughout the study, not just the time where you sit down and explain the study to them and have them sign that form. So it's important to think about when your informed consent form or formal consent process takes place. Are you handing them the research packet and then immediately turn around and saying, okay, do you want to participate? Or do they have some time to actually sit down and go over the consent document and ask questions? Another key thing with informed consent is it must be completely voluntary, and the participant must be able to withdraw at any time. And you need to remind them that they can withdraw at any time. This is kind of a legal technique called CRAM. That's a good acronym to help us understand, and if you ever had to demonstrate that the participant understood. So we use this a lot when obtaining consent for procedures and responding to different things. So the CRAM method involves communication of information, making sure the relative information is understood, the participant or patient understands and appreciates the situation, and they're able to manipulate the information you've given them and repeat it back in a rational manner. So I'm not telling you need to document everything the participant says, but kind of mentally think about, is the patient, first of all, literate enough in the language that I'm talking to them in? Am I using appropriate terms for their health literacy level? And are they able to convey this information back to me? It's something we kind of get in the habit of just going through the process. but it's really important with each and every patient to sit down and make sure they understand the information we're giving them. The next one is respect. So I know as many patients as a lot of us see, we kind of get in the habit of just going through the motions, but we do need to remember that we owe it to these participants who are giving up quite a bit to protect them. So we want to make sure that we treat them with dignity, privacy, confidentiality, and autonomy. So autonomy isn't one that comes up very often, but I make sure I add it into a lot of talks because a lot of times we just tell participants you have to do X, Y, and Z, and we don't think about that they are people with lives and have things to do. So we need to make sure that we're scheduling our study visits and things like that around participants' availability and making sure we're treating them with respect. So the next one we're going to talk about is benefits. So this is good for both the participants' perspective and us as researchers. We want to get the most bang for our buck, and we want to make the most out of a participant's sacrifice and get the most out of their data. So one of the things, or some of the things we can do to help with that is a power analysis, which we'll go over here shortly, continuous monitoring of our data, making sure our data is secure, and watching for those AEs, for those adverse events. Even if it's just a minor AE that's not study related, but we need to make sure we're keeping good track of those and following our reporting policies. So this is a question I get all the time, and I help quite a few folks with. What is a power analysis and things like that? And then the other response I get a lot is, I have to do hard math. No, importantly, folks out of my group, the biostatistics, epidemiology, research design group, or CORE, can help with this. So we can help you figure out about how many participants you're going to need to get a good effect size. And so we normally break effect size in a small, medium, and large. And we can either do this with theoretical data or actual pilot data. So I'm going to pick on Maria a little bit. She brought a proposal to me about an education paradigm, and she's like, well, how do I figure out how many people I need? I'm like, it's just simple math. And while this might not look like simple math to everybody, after a 5-minute conversation, I feel like we were able to figure out a rough estimate on about how many folks she needs to do that intervention with. And again, it can be something as simple as a casual conversation, and we can give you a pretty good estimate on how many participants you need. But with this, is it helps us get an appropriate number. So we're not recruiting 100 patients that we could have done a statistical analysis for and gotten a good p-value with 10 people. So it's important to do this, and not when we're writing RBs, just put, I'm going to recruit 100 patients. I'm going to recruit 250 patients. So it's always good to sit down and do these calculations, and we're more than happy to help you with this. The other thing that we don't think about a lot, and I see in quite a few publications and posters and things like that, are errors. So unfortunately, errors happen, but we need to do our best when we're designing our outcomes and our measures to make sure we're preventing these types of errors. So this is a interesting chart that kind of comically helps us figure out the 4 main types of errors. I'm sorry, the 2 types of errors compared to their correlating correctness. So a true positive is obviously the woman in this picture is pregnant, compared to the older gentleman. Here we get a type 1 error, which is a false positive, and then the other 2 is going to be the false negative, so telling the pregnant woman she's not pregnant. So this is always comical, but it helps to remember which types of errors occur when. And so we can do this through a bunch of different methods. The one that pops up to me first is doing our QI and QAs on our data. So making sure that measuring device has been quality controlled. Are these labs accurate? I know the folks down in the lab, we just send them blood and we get data back, but they go through a lot of processes every day to make sure those lab values are within acceptable limits. And the sponsor might not ask for it, but they can definitely provide you certificates or certifications that this machine is accurate and the sample is accurate within so many units of measure. So that's just something to consider. And then 2 more principles we need to go over real quick. Justice. So we need to make sure we're fairly selecting our participants. There is an even distribution of participants across different demographics to get a pretty good sample size. So this is a little bit of a headache here in West Virginia, since West Virginia is terribly diverse. And it's something I and a lot of other researchers have had to address in our publications and in our research proposals to the NIH and things like that. There isn't a huge Pacific Islander population, but the NIH wants Pacific Islander data, for example, in your proposals. So that's something that we need to think about, and it may have a big impact on our data. We may not be able to translate our findings really well to other study populations. But that's something we need to keep kind of in the back of our mind and do the best we can to help out and do, good science. Okay, the last principle I want to go over is non-malfeasance. So this goes back to, first of all, do no harm. So this goes back to watching for SAEs and things like that. We do not under any circumstance want to intentionally harm our participants. And this goes back to the voluntary withdrawal. If we have an SAE, we need to remind participants of the rights for things like preventative care or recursive care if something bad happened to them. And it's also important to be honest and upfront with your participants. So we also need to think about kind of do a risk benefit analysis. I know we do them a lot in clinic, but especially in research, do the risks of the study either outweigh or are rational with the potential benefits, maybe not directly to the participants, but at least to the population at large. And so a lot of our OMR, only minimal risk studies, don't have a direct benefit to the participant. And so we really need to weigh that risk against the potential benefit to the population. So with that, we'll take a quick break. Does anybody have any questions before we do a review game? Okay. With that, we're going to jump over. If you want to pull out your phones, and we will start a quick review game. You do not need an app for this. Let me get this shared. Share. So you can either go to www.kahoot.it.
You're not currently sharing.
Oh.
Hit share for me, please.
Ooh, there we go. There we go. Okay, so you can either go to www.kahoot.it or you can scan the QR code here. The important thing here is you do not need to put in your name. You can put in your favorite book character, your favorite movie character, a nickname. We're not taking quiz grades. I'll give everybody a second to get joined in. The fun thing is, this is one of my favorite tools to use. Is you guys will get ranked and scored based on how well you do and how quick you can answer the questions.
So I'm just going to pipe in here. I helped Tyler test this, and he tricked me because because the question was, I'm going to paraphrase, is Sally the best boss? Well, I wanted to beat him because I was behind. So the first answer was yes. So I hurry up and did yes. It was not the correct answer. It was heck yes or something. So I didn't read all the answers.
Yeah. Give you guys another couple seconds and we will start rolling. And give a bunch of I'm going to call it and we're going to start. Okay. Here we go. First question. True or false? A prospective study involves collecting new data. Very good. Looks like everybody got it right. True. A prospective study involves collecting new data. See our leaderboard here.
What's my name, Tyler?
Chuck Mullet. Which of the following is not a way to reduce bias? That one cut you guys off quick. I should have given you a little bit more time. We'll try on the next one. But correct, masking is not a way to reduce bias. What type of variable is changed on purpose? There we go. Let's move. A little bit of review here. Our independent variable is the one we change on purpose. So remember, our dependent variables are our outcome measures. So we depend on the IV, the independent variable. Categorical variables are the ones we kind of can't change, things like biological sex, blood types, and then confounding variables are things that might skew our outcome measures. So things like comorbidities, things like that. Ooh, we got a tight race here. What kind of control uses previously collected data? Very good. A historical control is the type of control that uses previously collected data. Nice. Which of the following is not a core ethics principle? I don't know who your PI is. It shouldn't.
It shouldn't, but
Correct. A patient outcome is not a core ethics principle. So we need to make sure we have respect for our participants. We're doing no harm. We're making sure we're doing the best by our participants. There's kind of a general, there's more benefit to the research than there is risk. So yes, patient outcomes are not a core ethics principle. What is the name of the test used to figure out how many patients we need in a study? Very good. We do conduct a power analysis to get a rough estimate of how many patients or participants we might need to do a effective statistical analysis. Okay. Non-malfeasance is the principle of I got some of you guys. It is the principle of doing no harm. We don't want to harm our participants. Yeah. Okay. And this last question is entirely so we can get some good feedback. It does not count for points. So, do I feel like this training was useful? There is a I prefer not to answer, and I will not be offended. Okay, I can live with that. And let's go to our podiums. Okay, third place is ESM. Second place is Stacy, and congratulations to NM, our first place winner. Just stop cheering. We'll do a couple case studies, and then I'll let you be free. Oop, nope, one. There we go. Okay, that was our review game. Hopefully everybody enjoyed that. We're gonna do 3 quick case studies with some pretty obvious flaws. If you would like, please drop a positive thing about the study in the chat and an obvious negative that you find, with an effort to be as productive and constructive as possible. So, case study 1, a research team is conducting a clinical trial to figure out if a new medication for chronic pain is effective in participants over 65. The participants were recruited primarily through online advertisements and local flyers with a link to a Discord chat website. The ads linked to e-consent forms, which required CAPTCHA and IP captures to ensure validity. The results of this investigation: the informed consent document was highly technical and not adequately explained to participants. Many participants had a limited understanding of potential risk and didn't understand that there was a placebo in the trial. So feel free to drop some of your thoughts in the chat, see if there's a good thing or a bad thing. Yeah, so somebody brought up a good point. Those of us who are more senior might have trouble working our smartphones, let alone operating a Discord. Yeah, Shelley brought up it's entirely too technical for this targeted population. Anybody think there was a positive? The one positive I would give these folks is they were able to send out a copy of their consent form prior to the participants coming in. Even if it wasn't a very good consent form, they at least gave folks some time to go over it. And to Cassandra's point, they did have multiple recruitment methods, which is good, even if they weren't very good methods for their targeted population. So think about this a little bit the next time you go to design your flyer or whatever. Make sure it's appropriate for your target audience. Okay. Our next case study. Last quick point by Mary. Again, it's too complicated. It might just discourage participants. You might not have a good recruitment goal. So case study 2, a research group is conducting a community intervention to see how local farmers markets might help reduce heart disease in a food desert. Results: most of the participants were from a similar demographic, and the results were hard to replicate in other regions. So what are your thoughts on this one? Okay, some of the good things about this one, I think it's a pretty good research idea. I think the group is probably good at heart. They're trying to see if they can use some local resources, like Sharon said, to help folks in the region. But I think the big flaw here is there's a pretty monolithic demographic, which makes it hard to translate to other populations. Yeah, it is a problem here in West Virginia too, but I bet you those researchers have a similar issue. A lot of our communities are pretty monolithic with some pretty unique socioeconomic and demographic problems, which may make this hard to translate to different demographic groups. Okay. We will jump to case study number 3. A psychology group is investigating a new anti-anxiety med versus a placebo. The participants and the researchers were not blind to the intervention. So that means the participants, the participants knew what they were getting. They were asked to submit their daily anxiety scores and other psychology metrics through a Google Doc. The result is the new medication had a significant side effect, but those in the experimental group reported both good and bad results because they knew they were getting the interventional product. And several participants accidentally saw other people's responses. So besides this sounding like a nightmare, are there any good things or any bad things you all think? Yeah, so to some of you all's point, Google Doc is not a very good way to share information, especially private information like your anxiety and mental health hygiene results with each other, let alone with other participants. That's a huge breach there. And the other thing is they tried to use a placebo control, but didn't implement a very good blinding system. So you might have been able to get away with single blinding here where just the participants didn't know. But they should have been pretty similar. So kind of the participants didn't know which group they were in. Nina brings up a good point. They did have an objective measure, so they did throw in some anxiety scales and psychological metrics, but yeah, definitely a hot mess. With that, I will turn it back over